In the previous video, you learned about the evolution of our sex chromosomes and about the X chromosome, more specifically. Now, let's focus on the Y chromosome. We left off our conversation about the X chromosome by noting that a very little portion of the X recombines with the Y chromosome, and this is called the pseudoautosomal region. There are only about 24 genes in the pseudoautosomal region, and otherwise X and Y do not recombine. This avoidance of recombination, it evolved because there is a gene on the Y chromosome called SRY. This is the sex-determining region in mammals and marsupials. Monotremes lack SRY. Their sex determination, it happens a bit differently from other extant mammals, but focusing on us. So the SRY region evolves quickly, relatively, and it is what sets off a cascade of events during embryonic development that leads to the formation of male phenotypic features. Now, because the Y chromosome has a gene on it, SRY, that is specific to males, its evolutionary history unfolded in a very different way compared to the X chromosome. Remember, the X chromosome is sometimes in a female body and sometimes in a male body, but the Y chromosome is only ever in a male body, and that has evolutionary implications. The Y chromosome is short and specific, and that's kind of the best way to think about the Y chromosome. Now, compared to the X, it has many fewer genes. In fact, the X chromosome's gene coding portion is 6 times longer than that of the Y chromosome. Only about 24 of the 1,094 genes on the X chromosome are also found on the Y. I apologize, I have a typo in the slide there. Ignore that. Okay, so only 24 are homologous. These are all found in that pseudoautosomal region. But aside from those, the Y has evolved to essentially avoid genetic exchange with the X chromosome. In a sense, much of the Y chromosome hides from the X chromosome. Now, because of this, over the last 300 million years, it has gained some new genes of its own, about 45 of them actually. Genes that code for proteins that are specifically useful to males. Any guess as to what those genes are involved with? What do male bodies do that female bodies do not? They produce sperm. All of those Y-specific genes are expressed in the testes, where sperm are produced. Over 300 million years of evolution, genes that benefit only males have been able to thrive on the Y chromosome. And so the Y chromosome is pretty much a short little chromosome that contains genes that exclusively serve male-specific reproductive features. Okay, so it makes sense that the Y chromosome is sort of a haven for male-specific genes. But why is it so short? And how did that happen? This phenomenon is what we call decay. There has been considerable decay in the non-recombining regions of the Y chromosome. Let's dive into why this decay has happened. All embryos start out female. 6 weeks after fertilization, they began to differentiate. The first trigger for this differentiation is the SRY gene on the Y chromosome. This gene starts this cascade of events that lead to the masculinization of the embryo. Initial recombination had to protect that sex-determining locus, SRY, so a barrier evolved to do so, and that barrier is a region that would not recombine during meiosis with the X chromosome. Now remember, for the heterogametic sex, there are 2 different sex chromosomes. Only one of those sex chromosomes carries the genetic trigger for male sex determination. The other sex chromosome is not male-specific. It's not female-specific either. Through generations, alleles that are very clearly favoring one sex over the other, they won't survive if they are on the X chromosome. And the SRY gene, well, pretty clearly favors males. So this is why it needed a barrier to protect it from recombination with the Y. So as soon as genetic sex determination started, you had this differentiation between the two chromosomes that came to be known as the sex chromosome. And the heterogametic sex, the one that is setting off the cascade of events that lead to the phenotype associated with the genotype of the heterogametic sex, that then had a very different evolutionary history than the chromosomes involved in the homogametic sex. Okay, so let's think about why the decay in the rest of the genes that are not on the X chromosome, and where on the Y, so think about the decay of the genes that would be on the X chromosome and that were on the Y chromosome 300 million years ago, but were lost through that process of decay. The decay is explained by the fact that the X chromosomes spend more of their time in a female body than a male body. So let's walk through this. So you have here a female in the pink oval and a male in the blue rectangle. So each of them has two sex chromosomes. We know that the male, the genetic male, has an X and a Y, and the genetic female has XX. So we have X1, X2, that's one female, X3, and Y1, and that is your genetic male. They have offspring, and so the female in the pink oval gets one X from mom, in this instance X1, and an X from dad, X3. They have a male child as well, so X2 comes from mom and Y1 comes from dad. Y2 mates with a female that introduces two new X chromosomes into this population, and then you mix up those chromosomes there, where you have X4 from the new female coming in, X2 inherited from the grandmother through the son, and then you have X5 coming in from the new female, and Y1, which is inherited from the grandfather, transmitted through the son. Now let's bring in another male. Another male from outside the group reproduces with the female, and here you have an X1 inherited from grandmother through the daughter, and you also have X6, which comes from that outside male. You have X3 inherited from, for the son, you have X3 coming from the grandfather, transmitted through the daughter, and Y2, which is inherited from the male who came in from the outside. Okay, so let's summarize this up. We'll summarize our pedigree here. We have 5 males, we have 5 females, we have 10 individuals, and we have 20 sex chromosomes in this pedigree. 15 of those are X chromosomes, and 5 of them are Y chromosomes. 10 of 15 X chromosomes that we see in this pedigree are paired with an X. So 2/3 of the time, an X is paired with another X chromosome. 5 of 15 X chromosomes are paired with a Y. So 1/3 of the time, an X chromosome is paired with a Y chromosome, and 2/3 of the time, it is paired with another X. So an X spends 2/3 of its time through the generations in a female. So alleles on X chromosomes that favor males, they can't actually get a toehold, as they'll be selected against when those alleles are in a male. They'll be selected for in a female body, possibly, depending on what they do, and they'll be selected against when they are in a male body. Now, any allele on an X chromosome that might favor a female to the detriment of males it just won't be favored because at some point it will be in a male body and it will be selected out of the population. But the Y chromosome, it is only ever in a male body, and therefore there can be significant conflict between X and Y in terms of what is positively being selected for, and remember, happening, passing down through the generations. Now, over evolutionary time, the Y chromosomes that had fewer and fewer conflicts with genes on the X chromosomes, those ended up being favored. So what we have found is that it ended up being more advantageous for a Y chromosome to just not try to compete with an X chromosome, because an X has got a lot going on. Sometimes lightly favoring females but can't actually harm males, and so it just ended up making for a better evolutionary strategy for the Y chromosome to just not compete. So all that we have left at this point in time is this tiny pseudo-autosomal region. Now, the drawback to the Y chromosome basically just not competing anymore with the X chromosome, or not having that recombination and the potential conflict there, is that people with one X and one Y chromosome are much more susceptible to recessive genetic diseases that occur in those 1,098 genes on the X chromosome. If you only have one copy of X, if your X has a deficient gene on it, you're stuck with it. This is why 8% of men are colorblind and just half of 1% of women are colorblind. Clearly, many women carry colorblind alleles, but they are not colorblind themselves so long as their second X chromosome has a typically functioning version of the gene to counterbalance it. Now, one more thing before we move off of the topic of genetic sex. Sex at the genomic level isn't just about X and Y chromosomes. There is also this phenomena called genetic imprinting. And genetic imprinting is when gene expression depends on whether the allele is inherited from the mother or from the father. There is differential gene expression. So genetic imprinting is epigenetic. It's an example of an epigenetic phenomena. This is parent of origin specific gene expression. The same genes are present, but how they are expressed varies depending on the parent from whom that version of the gene is inherited. Let's look at 3 examples. We're going to look at X inactivation, sometimes, Prader-Willi syndrome and Angelman syndrome on chromosome 15, and we'll look at Turner syndrome. Okay, X inactivation first. Remember earlier in this module, we talked about X inactivation in the example of cat coat color. Okay, the inactivation is random. About 50% of the time, mom's X is inactivated, and about 50% of the time, it's dad's that is inactivated. There are times when the X is inactivated because of which parent it was inherited from. So it's not random. Sometimes X inactivation is not random. For example, in the placenta. Oh, the placenta is so unbelievably cool. This is an organ that you made very early in your embryonic development so that you could attach to your mom's uterus. The placenta serves as the communication and transfer point between you and mom. Now, mom doesn't make the placenta, baby does. More specifically, baby's genome codes for the development of the placenta. Turns out that only the maternally derived X is active in the formation and function of the placenta. In XX babies, dad's X is always the one that is inactivated in the placenta. In XY babies, there's only one X and it's maternal, so no need to turn anything off. The differential expression of maternally versus paternally inherited X chromosomes is an example of X inactivation and also an example of genetic imprinting. And genetic imprinting is an example of epigenetics, aspects of gene expression that are inherited but are not actually coded for in DNA. Pretty cool, eh? Let's look at more examples. Let's look at a deletion that occurs on chromosome 15, near the centromere. So this deletion, it's not lethal so long as the other chromosome 15 has the region. However, the person who is missing this section of one of their copies of chromosome 15 does have an affected phenotype. There are some obvious consequences to not having two copies of this section of your genome, but the phenotypic effect, it differs depending on which chromosome 15 is functioning. When a person's only functioning copy of chromosome 15, of the section of chromosome 15, when that is from their father, the person's affected phenotype shows as having a developmental delay, sort of some speech impairment, puppet-like movements, seizures of laughter, and also they have some delayed cognitive development. This suite of effects, this suite of phenotypic effects, is called Angelman syndrome. When the only functioning copy of this section of chromosome 15 is from the mother, the person is unusually weak as an infant and has trouble suckling. By age 4 to 5 years, they have an uncontrollable appetite, so much so that families sometimes end up needing to lock cupboards and refrigerators so that the person cannot hurt themselves from overeating. These people, they tend to suffer from obesity and also show delayed cognitive development. This suite of phenotypic effects are referred to as Prader-Willi syndrome. Now notice how dramatically different the two syndromes are, and yet they result from the very same deletion. The only difference is which parent contributed the functional chromosome. Now, the genes in this region of chromosome 15 have an effect on how the body controls temperature, on the sensation of hunger, and on hormone production. This phenomena suggests that the epigenetic phenomena of genetic imprinting provides another source of variation in how hunger, hormone production, and other aspects of the hypothalamus's role in the body, of how that varies. So genetic imprinting can play a role in all of this as well. For the last example of genetic imprinting, I'll tell you about a study that was published in about 1995. In this study, scientists looked at 80 girls who have Turner syndrome. In this genetic syndrome, the person has only one X chromosome, and that's it. No other X and no Y. They have what we call monosomy, just one copy of the X chromosome and no other sex chromosomes. About 1 in 4,000 girl babies born has just one X chromosome. Now, without the presence of a Y chromosome, these people are, not surprisingly, phenotypically female because they don't have a SRY gene that would start that cascade of masculinization. Now, people with Turner syndrome, they tend to be a bit shorter in stature and their ovaries do not mature. They can also have some heart defects as well as some social adjustment issues. Of these 80 girls with Turner syndrome that were part of this study, 55 of them had an X chromosome that was inherited from their mom, and the remaining 25 had a paternally inherited X chromosome. The researchers conducted a survey of their behavior to see how the children managed in school and how they interacted socially in their families. The results showed that the girls whose X chromosome came from their fathers, they had significantly better verbal and social skills compared to the girls whose X came from their mothers. And so we see this effect on behavior that depends on the parent of origin of the X chromosome, whether it is paternally derived or maternally derived. And with that, we are now at the end of our investigation of genetic sex determination, but our investigation of sex has only just begun. We'll turn to anatomical sex next.